Okay, so continuing, just a reminder that a gene has more properties and factors than just protein coding sequences. Okay, so it's more complex than just the sequences of codons that code for the protein. So it has some other features that we will talk about and identify and explain in future chapters. So the first process produces a messenger RNA molecule through what we've called transcription, right? So we have the double-stranded molecule of DNA. It goes through transcription to produce an mRNA molecule. I've said this before and I'm saying it again. While the DNA has the information needed for the proteins and everything needed for the organism that we need, it is not active, meaning that we cannot use directly the DNA to understand within one process and create the polypeptide or the protein. So we first have DNA go through transcription, which product is an mRNA molecule or strand. The mRNA is readable and has the information that the DNA had, but in a way that the deciphering apparatus can work upon and read what we call a reading frame, right, to produce the peptide. In that process of producing the mRNA, we first have a non-mature mRNA molecule, and parts of that, of the RNA, have to be removed in order to produce an mRNA that we can work with. Okay, so the RNA molecule, which is produced from the DNA, has non-protein coding sequences that have to be removed in order to make an mRNA that is collinear with the protein to be produced. Once again, we have DNA, right? The DNA goes through transcription. We have RNA produced, but that RNA is not ready to be used yet. It still has non-protein coding sequences. Those sequences have to be removed, cleaved, in order to produce what we call the messenger RNA, the mRNA, that is collinear with the amino acids that need to be produced or added together, connected, to produce the outcome of the peptide that we need, okay, the protein that we need. So you're referred to a movie, enjoy. So the mRNA is actually a phase between the information only, meaning the DNA, the gene, and the actual production of the protein. It's in between. It's a mediator phase. The mRNA does not include all the non-protein coding regions that the DNA has. Okay, so it's shorter than DNA. The nucleotide triplets coding for amino acids in the mRNA and the DNA are the same, with the exclusion of the switch between T and U. So when we take the sequences that code for amino acids in the DNA, they are the same, besides that switch between T and U, as in the mRNA. The difference is that we took out everything that may interfere with the process of translation. Okay, so the 5' sequence going to the 3' sequence of DNA is the same as in the RNA strand, but it is not the same in the mRNA because we took out a few sequences that we did not need, and we call them non-protein coding sequences. Right? So RNA is longer, exactly like DNA, than mRNA. mRNA is shorter than RNA and DNA. So when a protein is produced from the mRNA, that process is called translation. And we will learn about translation in depth, exactly step by step how it's going to happen. So we have what we call a non-protein coding region at the start of a strand. It's on the 5' side, and it's called the leader. Okay? That region, also not exactly that region, but a similar region, meaning a non-protein coding region, also exists on the other side of the strand called the 3', right? And that is called the trailer. Okay? Just so you know. Non-coding translated regions means that they do not code for protein. In humans, transcriptions occurs in the nucleus, but the mRNA must first be transported into the cytoplasm for translation. Okay, so the whole process going from information, meaning DNA, right? Transcription in the nucleus, and then the last process of translation occurs in two different places. One in the nucleus, transcription, right? And the second in the cytoplasm, the part of translation. So the mRNA physically has to leave the nucleus, enter the cytoplasm, and there be translated. We can also understand that the actual production of the protein occurs in the cytoplasm. So we talked about immature mRNAs. That means that they have that phase that we have to activate them. That allows us to have mRNAs in a pool, or in a stack, and just use them when we need by activating them or having them go through maturation. So the process that makes mRNA mature is called splicing. The splicing is cutting out the non-coding regions that are still on the mRNA, okay? And certain changes that we will learn about to both the leader and the trailer. So we were talking about mRNA being a sequence that we can actually read. And we said that we need a deciphering apparatus that is able to look at mRNA, read mRNA, so to speak, decipher the codons, and give instructions how to combine and connect amino acids in order to create the right polypeptide or protein. And that apparatus is called the ribosome. Okay? It's a mixed apparatus. It has all kinds of units that we will learn about. It's very complicated, right? But very efficient. and it recognizes the codons, transforms them in a way to amino acids. When we look at the ribosome, it has certain components that are RNA components. So they are called ribosomal RNAs, or for short, rRNAs. Okay. We also have a certain type of RNA called tRNA, transfer RNAs. They are small units that what they know how to do is actually recognize the triplets together with the ribosome and incorporate. They're like taxis. They connect to the actual amino acid needed to be connected next. They incorporate it into the ribosome, connect it, and then the two amino acids are connected, and we'll go into that even more. So we have the mRNA, that is the information for the ribosome and the tRNAs. For what amino acids have to be added in the right order, and the ribosome connects between the mRNA and the tRNAs that are bringing the amino acids together. It reads the mRNA, tRNAs add the amino acids and are connected together, and now you have a protein. Okay, but we will go into that again and again and again. Right, so the ribosome is able to connect to the mRNA and function together in collaboration with the tRNAs to produce a protein, and you can actually see it as a factory of amino acid chain production. The gene actually includes the information for the tRNAs and the RNAs needed for the ribosome and the apparatus to work, and that's what we said, that those non-protein coding regions have other roles and other factors in them. Okay, so you're referred to a movie, enjoy it. When we look at certain sites, locuses that are located in the same DNA, we will call them cis sites or cis locuses. If the sites are not on the same DNA, they are defined as in trans. Okay, so we have different sites for certain aspects or traits or whatever we need done. If they are on the same DNA, cis. If not, they're in trans. Okay? And what we have is recognition factors that bind DNA at specific locations. Alright? If we have a mutation in such a special location that's supposed to be binded by recognition factors, it might cause the recognition factors not to be able to bind to the DNA. Now, recognition factors are important because they start the process of recognizing the DNA and where to start different processes. So a mistake there might completely annihilate the whole process, meaning that the gene will actually not be expressed, and that's not what we want, right? Basically, assuming that the gene has an essential and good related function it's in charge, and we don't want it expressed. But if the gene codes for a disease, the question is, would we like that to happen? Can a mutation in a recognition site prevent a disease? Why don't you answer that for yourself? But if you're confused, you can actually ask me and communicate that to me. So such binding sites for recognition factors are what we call controlling sites. They control the actual process. All right. Okay. We'll stop here and continue in the next and hopefully last video for this chapter.